to the right. So that's push. Down to all the way to the end when you're right. Yep. So wraps at the back. Thanks. Cheers. Right, just there. So it's not all the way to the end. It's just there. Yep. One back. Yep. 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 So it's just out to the right. Sorry, my bad. Not all the way. Hey, yep. Just through here. Sorry. Yep. Do you mind just at the back? Yep. Now with so many doors, we're going to go into this one here, which is another entry. And that's a pull as well. If I get you to hold, that'd be great. Thank you. Yep. Cheers. Is there a light in here somewhere? Just there to the right. That's all right, let's get that for you. Okay, we're going into another room. So we can all meet out here anyway. So just come in for now and... It does sound like reversing, doesn't it? Yeah. So where we are now is a microscopy facility. And if you look behind you, you'll see the ACRF Cancer Discovery Accelerator. There's a plaque. About 7 or 8 years ago, we received $10 million from the ACRF, which is the Australian Cancer Research Foundation. And that allowed us to get a lot of the equipment that you'll see. I mentioned $1 million piece of kit before. That's through the door there. We'll show you in a second. Where I'm going to go first is in here, and it's got a live cell confocal microscopy label. Unfortunately, the piece of equipment that we use for looking at live imaging of especially zebrafish has broken down for about the last 6 months. Right? But what I'm going to show you is just what it looks like, how you get into the machine, and then what the outcome of that is. Right? And so I can show you that on my computer. So this shouldn't be like a little violin playing here. It's really just an outcome of where we're at. The reason it's not working is this is an old piece of equipment that we can't actually get the computer to work for. The parts are fine, the computer's not registering it. So if there's any computer whizzes around here that can do things, we could really use some help. So we're going to go through. If you just hold that for a second, I'll get this here. That's the wrong one. That one.
Have you tried whacking?
Oh yeah, kick it a couple of times, I tell you. Yep. So if we come in, it's a tight room. So I mentioned earlier. Window. So this is the microscope here.
Wow.
So I would bet that if I asked you what that was, you wouldn't say it's a microscope, right? Yeah, because where do you even look at it, right? And so even in my time being involved as a scientist, this is where it's gone. We don't even get to look at the cells anymore down the scope. So this machine allows us to regulate temperature, so really finely controlled. The cells we put in there, or the animals we put in there, 37 degrees for cells. And Seb, what do we go, 28 degrees for fish? 28.5, right? And it really allows that fine control. It also allows us to put the exact amount of oxygen that you need, right? So whether that's 5%, 10%, whatever it needs to be. So that allows us then to put the animal or the cells in there and they're alive. We can then take images of them every 10 minutes, 10 seconds, you name it. This thing's actually called a spinning disc confocal. It's relatively new technology. So if you imagine that you're putting a laser onto tissue samples or cells, if you keep doing that constantly, it's going to get hot. It's going to get all these changes. So this has a disc that just spins. It has a hole in it. So this allows a little bit of the laser through so that you don't overexcite, overheat, and then actually affect the tissue. So I mentioned before, you know, prices. We got this for $200,000, right? It's been working for about 10 years. So actually it's a pretty good outcome. If we can get this computer to work, it's probably still open even, no it's not, then we'll be in a much better space. Manufactured in Japan, very thoroughly made. The way that you put your samples in is that this will, almost like a robot, will come out very precisely. We put our samples into this little cassette here, and that slides in and out. So samples go in here. This is where the gas exchange happens with these types of things here. And then that clicks into place, and then that should, in theory, slide into there very well. It locks into place there, and then that's how we put our samples into the machine. Yeah. So when we want to visualize the samples, you have to scan the entire area and then find out where your bits of tissue are or where your embryos are. So what do we get with that? It's a highly complex machine. It allows us to take a lot of images very quickly, right? And that allows us to do real-time imaging. And so what we get with it is, there's a lot of people around, if you can kind of squeeze in. So this is just this here is a zebrafish embryo, and it's gone for 12 hours. I don't know if you can see that. So each of those white spots is a neural crest cell. And we're looking at that because it's got a green fluorescent protein which is being excitated via UV light in that machine. We're taking images every 10 minutes. So what you can do is see that these cells start, and I mentioned before how neural crest cells move. See how they move so much, right? They start at the back here and go all the way into the front of the face. The cells that you'll see coming down through here are then going into these areas which we call pharyngeal arches. They're just areas which give rise to specific structures of the face. So we can see all of that in zebrafish. Why is that useful? We can actually trace each of those cells. We can watch how fast they move, where they go, do they live, do they die, right? And there's many measures we can actually do with that. In current science, we really want to be quantitative with everything we do. So this allows us the ability to say where all of those cells go, and that's color-coded over to the right. Does that make sense to everyone? Yeah. Now, we use this type of technology, but Seb's here, he would probably use different technology to this.
Well, we have a different microscope for imaging.
Yep.
But we have this for time lapse, which what Quentin has. So we're actually with the same fish over at Latrobe. They have exactly the same fluorescence. But I'm really impressed by this. I might have to get Quentin to do some of our work for us.
And so I've mentioned before, PhD. He started, the day he started, this broke down. And so we've also had to go through different ways of trying to get these real-time images, and it's been a complete nightmare, right? We're very hopeful this will become up and running. I can show you later, but we had to buy, fish don't need the high level of CO2, of oxygen regulated, they just need temperature. So we've had to buy a little heater to put on another microscope to keep them at the right temperature. Anyway, we'll get it there. Okay, so I'm going to move you now from this is called a spinning disc microscope, really good for real-time imaging. I'm going to show you this, the godfather or the grandmother of all of the microscopes we have. Hey. We're just going to go around down here. All right. Okay, so now this is one microscope in one room. All right. Different manufacturer. We had Yokogawa before. This is now it's called Leica. Some people that are probably enthusiasts of cameras would know Leica as a brand. So Leica make the microscope itself and all these other bits and pieces. I know it looks like you're probably, I don't know, in a Frankenstein movie here. All of those pipes going into the machine regulate the temperature, right? The temperature is really well controlled inside this box. The box then keeps us safe, because why this costs $1 million is this piece of equipment here. That's $500,000 just that. This is the lasers. And so we have lasers on the other machine you just saw, but this has lasers in a range that we can't see. So it's outside infrared. That allows us to do some magnificent techniques with this machinery. We can see now a single protein inside of a cell. on this one, right? So we looked at a whole organism before and cell migrating. This is looking inside that cell at what the proteins are doing. It's not easy to do, and you need the right techniques to do it, but that's where we're at, right? So cutting edge of what we can do with microscopy. It's actually called a STED, S-T-E-D. All right, so moving here to another type of microscope like this, not quite as powerful, but we can show you some images of some cells and actually have a look down the microscope this time. All right, so we're gonna come through.
And I'll probably just add to what Quentin said.
Yeah, do you just want to, wait, I'll go set up, yeah.
Is that the imaging capabilities? So way back when we could just look at a whole organism and we thought this is quite amazing, but now we can look at how the cells interact with each other. We can have cells of different colours as to how they work together. When we make our mutations, we can then see this is the effect this mutation has, not just on the cells, but on how they behave, how they move, how fast they move, how they form other structures. And so really, without this technology, we'd still be, our understanding would be much less than what it is now. But because the investment in technology and the imaging, and so all these different fields that come together really help us do our work as well, which then hopefully that's what going to translate to the clinic. So it really is impressive that we can go through this lab and see this, because this is not an opportunity many people get. But I think really to appreciate just how powerful being able to observe things really is, and just see how development happens in a model like the zebrafish, is what really drives research forward. How's that sound, Ralph? Is that
perfect?
sound good?
Yep.
Okay.
We're gonna move on to the other microscope room. Just right in front.
You guys want to-
Do you mind just passing that back and just put on, there's a little scanner outside that'll stop that beeping. Oh dear. So we're trying to get some really high magnification images, and for that we have to use different lenses. The lenses then need to be having no air interface with the sample, and so for that we need to use in this case it's a water immersion versus oil. Otherwise the light scatter happens in a way that you can't see the samples. Okay, so I'm really hopeful this is not going to cause us a problem. So we've talked before, thank you for that. We mentioned something called epifluorescence versus confocal on the slide. Epifluorescence is where light comes in and excites everything that you see, and then what you see is all of that light. A confocal microscope can then just say, I only want to see a very thin bit of the tissue, right? Or sample. I'll use an example of that. If I showed you this from above, you see everything, right? With a confocal microscope, I can say, I only want to see this bit in the middle. And then what would you see? You'd only see a ring, right? And this is what this allows us to do. So we can then reconstruct an entire bottle in this case and get that in a three-dimensional reconstruction, right? That comes with a lot of costs, right? Because then you have to actually specifically define where you're looking. Say that again, sorry? Yes, yeah, exactly, yeah. We're going to show you a CT image in a second. So CT works on the fact that you get a X-ray and then you rotate X-ray, and you do that a million times in a 360 degree. And then you can actually computed tomography used to actually make a 3D reconstruction of it. It really helps with a craniofacial development because it's such a like a three-dimensional type of process, and the morphogenesis is really hard. Excellent. We're back working. So I'm hopeful of showing. I can show you pictures on the computer, but it looks so much better down the microscope. Whoa, that was a bit rough. Okay, so apologies for this not being set up already. Okay. All right, hopefully you'll be able to see this under here. And if I go to...
Looks like opal.
Yeah. What do we want to see here? What I'm doing, I'm telling the microscope what channels and what colours I want to see. The blue is something called DAPI, which is the nucleus, which is just here. I'm going to go to fastest. I don't want that. I want green. Ah, dear. Okay, that's fine. So it already takes, weeks of time just to learn how to use this piece of equipment. And then when we get a new piece of equipment, you've got to relearn it again. And it's, I guess you'd appreciate that. It just takes a long time to become experienced with it.
And the hardest part's probably unlearning what you already learned.
Yeah.
You have to go digging for them. It's not a gem.
All right, now I'm just changing. I can see blue down here at the moment. I'm just changing that to green. Yeah, excellent. All right, so I'm going to leave it on green because I think it shows you more of what I want to see. But I would like, if you want to have a look down the microscope, then please do. You just have to look down. It should be in focus, and what you're looking for is some green cells. If you want to. What we're looking at is something like this. So we mentioned before, so Raf's project, who's behind here, we've removed a specific gene, and someone asked about removing genes before. We've specifically removed vascular endothelial growth factor, VEGF, from neural crest cells in this case. And, well, actually, these are cartilage cells. I apologise. And this is what the normal ones look like. So we have, can you see the blue? Right, the blue is the DNA and the nucleus. I can get rid of the other channels. That's the nucleus, where all the DNA is kept, right? We can put another antibody in this case to look at the Golgi network, and this is where, specific things happen within cells. And that's those white bits in each cell. You can see that little, almost like ribbons. You can see that. Maybe I can make it a bit bigger, actually. Anyway, and then the green is something called the endoplasmic reticulum. This is where all the proteins get changed so they can go out and do their functions. So this is a normal cell. That's what they should look like. Now we get rid of vascular endothelial growth factor in these cells, and what do you see happen to the green? So if we go side by side, here it's very structured and spread out, and then hopefully what you'd see here is it's all spotty. Yeah, that happens when the cell becomes stressed. Yeah. Yeah, so I'll zoom in a bit. There, that's one cell, right? And if I go back here and we zoom in a bit more again, maybe we'll look at this cell here. That's the nucleus of a cell, that's the out parts of the cell, right? And so...
And that cell's not stressed.
That's not stressed, yeah, right? And then when we get rid of just one gene, that's what happens. Right, I think even this one here, you'll probably see the nucleus is starting to get a bit funny as well. Like it's spreading out. And what we know is that if we leave that go, these cells will die. So by knowing that that's stressed and it's the endoplasmic reticulum, it tells us what pathway is affected. We think it's because there's not enough amino acids. And that's why I mentioned before about we add one amino acid to the drinking water of the mice and it helps, right? We still don't know if that's true. Right. We know the mice it helps, we don't know if it's amino acids that do it. So we're still trying to do that. But if you want to have a quick look, please do. You're looking for something like that under here. Hopefully you can see it, but
So, this is probably a really dumb question,
there's no dumb questions.
because I know absolutely nothing about medicine or science. But I always assumed that cells are all the same size and same shape.
Yeah, no.
They're not?
I'm going to show you something in a second. Hopefully, it'll blow your mind.
Stop laughing.
Right. So just for example, we look at one extreme, which is a neuron. You have neurons sitting in your spine here, which send an axon all the way to your toes,
Right.
and that's called the sciatic nerve.
Oh, okay. Yeah.
So one cell has that much length in it, and that's why we all have bad problems with sciatica, right? So I'm struggling with it. I'm 49, and so you'd know over time we don't walk the same. Our gait gets different. We always fall over. This is one of the most prone nerves to having effects. Yeah, but then you look at. You'll see a neuron in addition a second compared to precursors of them, and they have these beautiful, lovely extensions on them, and yeah. Yeah, and then.
I'm pleased to think that I look like that inside.
I would say that this is probably not the most visually interesting thing to look at. So if you want to hold off, I've got the best of all is on the dissecting microscope upstairs. If you do want to hold out for that, it'd be a bit easier to see. You can actually see it like physically. Yep. It's a bit. Yeah. It's not the greatest of all. Yeah. All right. Yeah, it does take a little while for sure. So if you want to, we'll move along. I'll just get my cheat sheet of where I'm supposed to go, which now I've probably lost. Oh, there it is. Thank you for that. We're going to go to the dissecting microscope. Thanks, Raf. So, Raf, I'd go right. Oh, yeah. Yep. The other right. The proper right. Could you see them?
Yes.
Yep.
It's fascinating. Absolutely fascinating. Thank you.
Thanks for that.
This is really cool.
I think it's phenomenal
Oh,
how far science has come.
one of the questions that Ben was going to ask me, which I just talked too long about other stuff, was like new technologies.
Yeah.
Now, if we press up, that'd be please. Yep. Press on. Is it? There's only one. We may all fit in here. This is a service lift. I think we should all fit. Yep. Yep.
Is there a drink service?
No, just want to scan this over here. Thanks. So I'm now taking you into our main lab. So most of my people work on level 10. We are expanding out to level 8 where we're creating a stem cell facility. That's where we're taking those cells and trying to push them into facial elements and things like that. So we're going to go around to the right. Yep. Hey. I've got there's 700s on. It doesn't look so good down the microscope, but the pictures are there. Yep. Just going to get Raf to set us up with the next thing. We're going to come all the way to the end here. This time all the way to the end properly, yeah? Okay, so this time we're at a really low resolution microscope. But the samples are, in my opinion, are spectacular. All right, so what we have here, and apologies if there's some squirmishness, but this is called a dissecting microscope. We can only go up to at a maximum 6.3 times magnification, and we can see the image here on the right. Right, so what I would hope you can see.
So what is that?
Yeah, so this here is an embryo from a mouse that we've taken from a pregnant mother. We've fixed it and then stained it with things that bring up bone and cartilage. So bone here is red or dark purple, and the cartilage, the chondrocytes, are blue. Right? I said before that in this case, the jaw especially forms with a chondrocyte precursor called Meckel's cartilage, and the bone comes in and wraps around it and then forms that our jaw. Right? So on the left-hand side, we've got a normal mouse embryo. If you come up close, you should be able to see it anyway. You can just see them there. There's the ribs, there's the head, the tail. I would love it if you can have a look under here, because that doesn't do it justice. Right? So we'll start low magnification, and what we've got on the left is a normal wild-type mouse. If you can see here on the picture, that's the jaw length that should be there. Now we've got a mouse, and Seb mentioned before some good microdontia. This one here, I hope you appreciate it, the jaw's a lot smaller, right? And that's from one gene missing, right? That's that VEGF gene we're talking about, right? And so that is, a sense, a model of small jaw that kids often have as part of their craniofacial defects. We're starting to learn why that's the case, and that's what we're trying to fix, right? So I'll put it back over here, and then have a. It looks, that does it anyway. You have a look yourself. So if we look down here, do you want to have a look yourself?
Yeah, okay.
Yep. It should be in focus.
How developed are they?
Yeah, so this is about 17.5 days past, conception, yep. It'll be born a day after this. Yeah. Yep. Could you see that?
Yeah.
Looks better down there.
Yeah.
Right. So clear. Yeah. Can you see that?
Yeah. It is clear.
Right. It's so much better from looking down the scope. So please all have a look. I mean, it's, yeah.
So, I gave a talk in Perth last week,
Yep.
2 weeks ago, and I actually used these images of yours
Oh, okay.
and to reference.
Yeah. Raf, can you go turn the Zoe on? Can you get some stem cells out, neural crest cells, chondrocytes? Excellent, thanks. Do you agree with me that it looks much better down there?
Oh, yeah.
Yeah.
Oh, wow. That sort of looks plastic.
Yeah, two-dimensional, and you just don't get it, right? So, yeah, yep. So there's still a lot of things that need to happen in this mouse. So we talked about craniosynostosis before. That's these elements up here, which you know that children are born with fontanelles and they're not shut. That's what these those gaps are up here. The mouse skull, I think, only fuses at about 2 weeks, 2-3 weeks post-birth. So that allows room for expansion of growth and then of the brain formation as well.
That's incredible, isn't it? Once the skull has fused, there's no way of unfusing?
Yeah, so for craniosynostosis, one of the therapies is actually just to go in and physically break it, right? To cut it out and remove it, and then allow that to happen, to expand. There's some really great work done down the road here at SAMRI via Stan Gronthos's group, and they've been working on ways to try and stop bone growth so that they could actually, physically stop it, not physically stop it, using just factors injected, right? So that's ongoing research they're still doing. There's many areas. I mean, I'm very specific in what we do. Seb's working on his, different sets of genes. Yeah.
Yeah, I think what Quentin said before about we know an absolute lot about very little is very true. So we're, like international experts in a very, very tiny area that no one cares about except the other international experts.
So dinner
that.
parties would be very quiet once you got through your
Oh
topic.
no, we always
Yeah, good.
we can always talk about other people's work, but that's what we love doing. That's what we do. So we're always learning. I think that's what we do as scientists. We never stop learning.
Can I move it across to the other one?
Of course, yeah.
Because I find that fascinating. And that's one gene.
One gene.
One gene difference.
Yep. And the power of this is actually even, you know how we said we can make these mutations in the whole of the genome. This is only in the neural crest cells. Every other cell in the body has that normal gene. So it's a powerful technique we can use, right?
So, in a mouse, would that mouse survive, or would it have such problems that it couldn't feed properly?
We have let them go, and they don't feed.
They don't.
They don't survive. Yep.
Yeah, that makes sense.
Yeah. Has anyone else want to look?
Use that research to actually stop mass spikes.
Yes. Yeah. Anyone else need to look? We're going to move around now to another area, and this time we're going to look at cells. So different types of cells. We'll look at stem cells. We'll look at neural crest cells and more mature chondrocytes. Have you got them? What do you got there? Stem cells?
Yep, stems.
Brilliant. Okay. Yep. Thank you, Raf.